In this video, we will cover mitosis, meiosis, and the cell cycle. We will look at the key stages of mitosis, the key stages of meiosis, and the differences between the two. Now, both mitosis and meiosis are means by which cells can reproduce themselves. And depending on what cells we are looking at and what cells need to be copied will determine whether mitosis or meiosis occurs. Now, with regard to the cell cycle is drawn as a circle. And it is drawn as a circle because a cycle is a repeating set of events. So the cell cycle is a repeating set of events that compose the life of a cell. And we're going to take the cell cycle and divide it into two periods, interphase and cell division being commonly called mitosis. Here we have the diagram of the cell cycle. We're going to go with the cell cycle lasting 18 to 24 hours for completion. Do remember that a cycle indicates a repeating pattern of events. And so with the cell cycle, we are looking at the events that lead to the reproduction of new cells from old worn out cells. And as we look at this diagram, you will notice that from right here all the way around to right here, we have interphase. Interphase is the time between divisions, and interphase is divided into three phases. The G1, which would last about 10 hours out of a 24-hour period. The S phase, which would last 9 hours out of a 24-hour period. And the G2 phase, which would last about 4 hours in a 24-hour period. The G1 phase stands for first growth, and here we have the offspring cells growing to mature size. The S phase represents the synthesis phase, and it is in this phase that DNA is copying itself and going through DNA replication. The G2 stands for second growth, and this is the gap after the DNA synthesizes, and it's just before division. So here the cell is preparing to divide. Now, the actual cell division itself lasts about 1 hour in a 24-hour period and would be right here represented in green, with cytokinesis being represented right here in the red. As we look at the stages of mitosis, we can learn the stages in order by learning the word IPMAT. IPMAT stands for interphase, prophase, metaphase, anaphase, and telophase. Now, when we begin with interphase is not a true stage of mitosis. However, we always discuss interphase when we talk about cell division. Interphase is composed of three stages, G1, S, and G2. And so in interphase, it is commonly said that the cell is resting. However, the cell is doing everything but resting. Here we have the cell growing, we have DNA replication occurring, and we have the cell preparing to divide. We can identify interphase by a clearly defined nucleus. And this will be the first stage and the only stage that we're going to see a clearly defined nucleus. On average, the cell will spend usually 90-95% of its time in interphase. Moving on, the first true stage of mitosis is prophase, and prophase can be subdivided into early prophase and late prophase. In early prophase, we start to see the chromosomes begin to coil up. Now, when the chromosomes are uncoiled, they are called chromatin, and so in interphase, the chromatin is located in the nucleus. However, in prophase, the chromatin has begun to coil and become visible as chromosomes. The nucleus has broken down, and since the chromosomes have coiled up, this will look very spaghetti-like, with the chromosomes being readily identifiable. This is the longest phase. The centrosomes with the centrioles will begin to move to opposite sides of the pole. We can see that here as they move to opposite sides. And then we see the spindle fibers begin to radiate away from them to attach to the center of the chromosome, known as the centromere. Moving to metaphase. Metaphase is identified because the chromosomes have lined up in a straight line in the center of the cell. The center of the cell is known as the equatorial plane. Now you'll notice that our centrioles are on opposite sides, that our spindle fibers have attached to our centrioles and are attached to the center of the chromosome, known as the centromere. In anaphase, anaphase is one of our shorter stages, and so when we look at anaphase, we have seen the centriole begin to wind in the spindle fibers, and as it winds in the spindle fibers, that will pull half of the chromosome in this direction and half the chromosome will be pulled in this direction. Now, because the chromosome is being pulled, this is often described as look like it's wiped out water skiing. And we say that because we call our centriole a boat, and then we have our spindle fibers that would be like ropes, and then we would have our little skier here who's wiped out and his little legs are flying behind them. And so what we're seeing is half the chromosome moves to this side of the cell and half the chromosome moves to this side of the cell. Now in telophase, what we have happening is the spindle fibers will release from the chromosomes, and we will start to see the chromosomes unwind to make chromatin. The nucleus will begin to reappear. If this is an animal cell, we will see a cell plate, we will see cleavage furrows, which is this pinching off area. If it's a plant cell, we will see a cell plate beginning to form that will separate the two cells. Now before we move on, I do want to stop and go over the difference between chromatin, chromosome, and chromatid. When we have unwound chromosomes, such as we have present in interphase, we call those unwound chromosomes chromatin. A chromosome, that's indicated by this photo and this, looks like an X. And the center part of the X is the centromere, and attached to the X we have half of a chromosome, and we have this half of the chromosome. And half of a chromosome is known as a chromatid. So to make up a chromosome, we have two sister chromatids attached at the center, known as the centromere. The entire structure is a chromosome. Unwound chromosomes are called chromatin. Half of a chromosome is known as a chromatid. Moving from mitosis and now looking at meiosis. Meiosis is a process in which gonad cells, or sex cells, divide twice to produce haploid cells. Haploid cells carry half the chromosome material. So in humans, the haploid number would be 23. Mitosis carries the complete chromosome number, and that's called the diploid number. In humans, that would be 46. The reason for the twice division of our gonads is so that each sex cell can carry half of the genetic material. This allows for each species to maintain a set chromosome number. So the cells preparing for meiosis will first undergo the G1S and G2 phase of interphase, just like in mitosis. And then we have meiosis 1 and meiosis 2, and these are named for the two divisions of meiosis. There are some important differences in the stages compared to mitosis. And so as we look at these stages, meiosis 1 has 4 stages. It has prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, this is similar to the mitotic prophase except for synapsis. Synapsis is where the homologous pairs of chromosomes pair up and twist around one another. Tetrads is the term that's used for these paired homologous chromosomes. Crossing over occurs, and this occurs when part of the chromatids exchange the genetic material. We'll talk more about that on our next slide. Moving to metaphase 1, the tetrads line up randomly at the middle of the cell. The spindle fibers from one pole attach to one centromere of one of the chromosomes. The spindle fibers from the other pole attaches to the other chromosome. So here we have the chromosomes being attached. They will not be separated. Now if you remember on mitosis, when they attached in anaphase, we had the pulling apart of the chromosomes. This is different from anaphase 1 of meiosis. The spindle fibers will randomly pull the entire chromosome to a separate pole. This is an independent assortment, and so it's completely random. In telophase 1, this is the final phase of meiosis 1, and the chromosomes will reach their opposite sides. Cytokinesis will then occur, and so we see the separation of the cytoplasm, and this will make two new cells. At this point, the two new cells contain haploid numbers of chromosomes, yet each has two sister chromatids attached to it, and so each chromosome is still attached. On our previous slide, we mentioned that crossing over during synapsis occurs. Now, beginning with synapsis, our homologous chromosomes will pair up. Next, what happens is that they twist around one another. And then the actual crossing over occurs. And this is where parts of the chromatids exchange genes. And so we can see that exchange here, and we can see that exchange here. Now the result of this is going to be genetic recombination, and this results in an increase in variation among species. So quickly reviewing over meiosis 1, we begin with interphase. Now with interphase, we have the same three divisions going on, and so we will have the G1S and G2 phases of interphase. When we get to prophase, prophase 1, the chromatin will have wound up to form chromosomes. The chromosomes have become visible, and so we have our nucleus breaking down, the crossing over occurring, and the chromosomes becoming visible. In metaphase 1, we have the two sets of chromosomes lining up. So instead of having a single line of chromosomes, we have two lines of chromosomes. And so we say that the tetrads have lined up randomly in the middle of the cell. In anaphase, what we have occurring is that the spindle fibers pull the complete chromosome to one side of the cell. So we will have 23 chromosomes being pulled to this pole, and we'll have 23 chromosomes being pulled to this pole. So that when we reach telophase, in telophase, we have the cleavage furrow forming, we have the cell pinching off, and so that we have 23 complete chromosomes on this side and 23 complete chromosomes on this cell. So we have two cells formed. They are not identical, each cell containing 23 chromosomes. So at this point we say that the cells are haploid, meaning they contain half the chromosome number that would be found in that species. After telophase 1 has been completed, we move to our second division, and our second division would be prophase 2, metaphase 2, anaphase 2, and telophase 2. Now the key thing for meiosis 2 is that the DNA is not duplicated, and so we do not have an interphase occurring. Now in prophase 2, the spindle fibers will form and begin to move the chromosome to the middle of the cell. In metaphase 2, the chromosomes are lined up in the middle of the cell, and we have one single line. We have the spindle fibers attaching to the chromosomes, so that when we reach anaphase 2, the chromosomes are being pulled apart, with half a chromosome, a chromatid, being pulled to each pole. And so we will have 23 chromatids pulled to this pole, and we will have 23 chromatids pulled to this pole. Now remember that from telophase 1, we formed 2 cells. So when we go to prophase 2, we have 2 cells going through this division. Metaphase 2, we have 2 cells going through this division. In anaphase 2, we have 2 cells going through this division. And so we reach telophase 2 and cytokinesis. Now when this happens and our cell pinches off, we will have each cell pinching off to make 2 cells. So since we had 2 cells going through this portion, we end up with 4 haploid cells for each one cell that began the meiosis cycle. Here we see meiosis 2, and notice that because telophase 1 produced 2 cells, we will see both cells going through meiosis 2, and those stages will be prophase 2, metaphase 2, anaphase 2, and then telophase 2 with cytokinesis. In prophase 2, we see the nucleus breaking down, and we see the chromosomes becoming visible. They will begin to move towards the center of the cell, and you'll notice that they start to line up in the center of the cell for metaphase 2. And the spindle fibers will attach to the centrioles and to the centromere portion of the chromosome. In anaphase 2, we see that half a chromosome is being pulled to this pole, and half a chromosome is being pulled to this pole. And so what we see in prophase 2, metaphase 2, and anaphase 2 is almost identical to what we see in mitosis. Finally, with our telophase 2, we have that pinching off occurring, and so we have 2 cells formed here, 2 cells formed from here. The cells are not identical. They are haploid, meaning they carry half of the genetic material. So to sum up mitosis and meiosis, mitosis is the process of asexual reproduction in which the cell will divide to produce 2 identical cells, each with a diploid number of chromosomes. That means that each cell is identical, containing 46 chromosomes. Meiosis is cellular reproduction to form gametes, and so the number of chromosomes is reduced in half, and so we have the haploid number of chromosomes, and this allows for when we recombine the gametes during sexual reproduction, then a species would maintain a constant chromosome number. So mitosis is an asexual type of reproduction. Meiosis is a sexual type of reproduction. The purpose of mitosis is to replace worn out or damaged cells of the body. The purpose of meiosis is to produce gametes for sexual reproduction and to maintain a constant chromosome number within species. Mitosis occurs in all organisms within the somatic cells. That means every cell in the body with the exception of the gametes, the egg and the sperm. Meiosis occurs in humans, animals, plants, and fungus, and it only takes place with regard to forming gametes, or sex cells. Mitosis goes through 1 division, ipmat, interphase, prophase, metaphase, anaphase, telophase, interphase not being a true stage of mitosis. Meiosis goes through two divisions, interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. In mitosis, we will form two identical cells. In meiosis, we will form 4 cells that are not identical, and they are haploid in nature, carrying only half the chromosome number. So in humans, these would carry 23 chromosomes, where in mitosis they would carry 46. There is no mixing over or crossing over in mitosis. However, crossing over is extremely common in meiosis. Here we have the same number of cells formed as the parent. Here we have half of the original gamete cells before meiosis.